Hey everyone, welcome to the BGE Navigator tutorial series. In this video, we're going to do a deep dive into the Taxonomy selection tab. This is where your analysis journey starts. You will learn how to navigate the cell type hierarchy, understand the assay indicators, use the search function, and explore region distribution charts. Let's get into it. So let's click on the Taxonomy selection tab. This is the first step in the 3 steps workflow: taxonomy, assay, and browser. The idea is that you narrow down which cell types you are interested in before you start picking tracks. The taxonomy data comes from BICAN Consortium and is organized hierarchically. At the top level, you will have your neighborhood. These are broader category of brain cell types. Under each neighborhood, you have class, then subclass, then finally individual groups. At the very top of the taxonomy panel, you will find the assay type legend. There are three assay types and annotation type, each representing a different set of modality and display its own set of cell types and available data. Now let's look at the tree structure. Each neighborhood appears as a selection header. Click and expand the arrow to open it up and see what class and subclass and groups are underneath it. On each row, you will see small colored dots. Each dot indicates one data availability for that assay. Hover over any dot and you will get a rich tooltip card. It shows the assay name and the list of available modalities. The colors are from colorblind-friendly palette, so each assay gets its own distinct color. There is also another legend showing which species we have data of. You can also use the expand all or collapse all to quickly open or collect the entire tree. Now for the actual selection. There are several ways to select cell types. You can select subclass or group individually, or you can select all group underneath any subclass. Or alternatively, you can use the select all button right here to select all the taxonomy terms, or clear all to clear the selection. If you know what cell types you are looking for, the search bar is your best friend. You can search terms like MSN, and the tree will automatically filter and expand to show the matched entries. The search works across all the levels. It matches against the neighborhood name, class name, subclass name, and group names. To clear the search, just delete the text in the search bar and the full tree will reappear. Now let's scroll down below the taxonomy tree. You will find the region distribution analysis section. This is a feature that shows you where your selected cell types come from in the brain. For the subclass selection, on the plot on the left shows you the cell are harvested from which regions, and on the right it's group region distribution, showing the same information. Scroll down further, and you will find this region abbreviation legend that maps each abbreviation to its full name. Also, at the bottom of the taxonomy page, you will find Brain Anatomical Structural Explorer. These are species-specific buttons that open interactive atlas views from both the Allen Brain Atlas and the Scalable Brain Atlas. They are embedded within the portal, which help you understand the anatomical structures associated with the brain regions. When you are done selecting cell types, click the Next button at the bottom of the page. This takes you to the Assay Selection tab, where you will see all the tracks matching your taxonomy selection. Your selection are preserved as you navigate between tabs, so you can always come back here and adjust. That's the Taxonomy Selection tab. In the next video, we will cover Assay Selection in detail. Thanks for watching.